Welcome back, ladies and gentlemen. Today I'm going to talk to you about homeostasis and transport. So let's break these words down before we get started. Homeo means same and stasis means state. So homeostasis is the ability of an organism or a cell to maintain the same state. We'll talk about that more in a few minutes. We also have trans, and trans means across. So transport is the ability to move a material across a space. We have a quick little review. What serves as a barrier between the inside and outside of the cell while controlling what comes in and out of that cell? Well, hopefully you're thinking about your egg lab and this egg that had a coating around it that prevented certain materials like syrup from going through it, but allowed other materials like water to pass through no problem. So what is this outer coating called? The plasma membrane. The movement from high concentration to a low concentration, we have talked about very little, but imagine that I were up here and sprayed an air freshener in the room. There's going to be a high concentration of those smelly good chemicals near where I sprayed the air freshener, and throughout the room we have less of those chemicals. So that movement across the space of those smelly good chemicals is called diffusion, and this works with all kinds of materials, not just air freshener. So does diffusion take energy? Well, do I have to stand up here and fan my air freshener to make sure that it gets throughout the whole room? No, okay? So these are some of the topics that we're going to be talking about in detail today. Here we have homeostasis again for same state, and this is the ability of a cell or an organism to maintain a stable internal environment regardless of what's happening outside of that cell or organism. And homeostasis or balance must be maintained in order to support life. Life requires these biochemical reactions to happen at speeds fast enough to support it. So we've talked about some molecules already that lower the temperature that reactions can happen and allow them to happen at body temperature. And these same molecules also allow these reactions to happen faster than they normally would outside the body. What were those molecules called? Hopefully you're thinking proteins and enzymes, right? Enzymes are certain proteins that catalyze reactions or make them happen faster. Now, cells do require certain conditions, and some cells have conditions that are different from other cells. For example, the cells in your stomach need a very low pH of about 2. But if this pH existed in your mouth or on your skin, those cells could not survive. They need a pH of around 7 or neutral. So just a little example there to help you remember that not all cells require the same kinds of conditions. Certain things that must be maintained in an organism, and there are many others, but pH, we already kind of talked about that, glucose levels, water balance, and temperature. So if your pH gets off balance in your stomach, say it gets too high or too low, then you might take something like Tums or Rolaids in order to restore that pH back to where it's supposed to be, or you're going to be very uncomfortable. If glucose levels get out of balance, then you can have diabetes, right? And this is an example here of a young lady getting something called dialysis. And dialysis takes out the blood in the body and runs it through this machine. And it's basically cleaning the blood of uric acid and creatine that's going to build up in the blood when glucose levels are off balance. Water balance must also be maintained. And we can think about our case of Jennifer Strange. We can think about our eggs, right? Now, water balance has to be maintained in order to stay properly hydrated also, so that the cytoplasm has the right amount of water in it for these chemical reactions to take place. If your temperature gets off balance or out of homeostasis, then you can run a fever. And remember, we talked about some of the benefits of running a fever to destroy those bacterial enzymes and kill the bacteria that are making you sick. But again, you would not want your fever to get too high because that can destroy your own enzymes and lead to death. We use a pH scale to describe how acidic or basic a solution is. So neutral would be 7. Bases are above 7. They have a lot of hydroxide ions. And acids are below 7, and they have a lot of hydrogen ions or positive charges in that solution. You can see that your blood is a pH of around 7.4. And this pH can affect the rate of these biochemical reactions. We saw that with your enzyme lab that you did online. Now, I talked to you about those Tums and Rolaids that you might take if your stomach acid gets off balance. And those would be examples of buffers. Buffers are used by cells to maintain a certain pH. And buffers are mixes of acids and bases, and they can take in hydrogen ions or release hydrogen ions in order to level this pH off to where it's supposed to be. So when you add an acid or a base to a solution that has been buffered, the pH is not going to change because that buffer, again, can take in hydrogen ions and bring that pH back toward neutral, or it can secrete hydrogen ions and bring that pH back down toward an acid. When we talked about cell organelles, we talked about the plasma membrane, and the plasma membrane controls what comes in and out of the cell. We saw this with the egg lab. This is essential to the cell's survival, that it can control what passes from the outside to the inside and vice versa in this cell. So it is made mostly of phospholipids. You can see one phospholipid here that I'm circling with my cursor. We have a phosphate head and two fatty acid tails. Those phosphate heads are hydrophilic. They like water and don't mind being around it. And that's good because we're going to imagine seeing a lot of water on the outside of this cell and on the inside of this cell when it's in contact with the cytoplasm, which is made up mostly of water. Our cell membrane is also made of proteins scattered throughout it. You can see some of those here. And these proteins can freely move around in this cell membrane. They are not stuck in these phospholipids. So to imagine this, when we did the cell lecture, I told you to imagine a pool full of water, and on the surface of that water, we packed as many of those little plastic playpen balls that you used to play in as a kid together as possible. And I told you to imagine dropping a penny through those little balls. And you can kind of see what I'm going for here with these phosphate heads on this phospholipid bilayer. But if we imagine dropping the penny into the pool, it's going to squeeze right through the holes in those balls where they're packed up tightly together. Now, big things like a notebook or a piece of paper are not going to pass right through those balls. They're going to sit there on top. In order to get that to pass through, I told you to imagine getting into the pool and pushing those balls aside and opening up a hole or a channel to the water. And that would allow your big thing to come through. So that's kind of what's going on with our cell membrane. Small things that have no charge can pass right through the little holes in this phospholipid bilayer. But large and charged molecules are going to be repelled by these phospholipids, and they're going to need a hole, like a protein channel, to go through. So when we think about what happened with your eggs, your water egg, there was more water outside of the cell than inside, so the water's going to want to flow through. Now, water does have a charge, so it is going to need a protein channel to move through this cell membrane. So water moves into the cell, but when we look at our syrup egg, it doesn't matter how much syrup was on the outside of the cell. The syrup did not go into this cell. The water was instead sucked back out. Syrup is too big to fit through these little phospholipids, and there are only certain proteins that allow sugar to pass through a cell, and our egg cell has those shut off, and it's not using them. So that's why your syrup doesn't come through. So once again, small uncharged things go through the phospholipids, and large and charged molecules will have to go through a protein, and phospholipids and proteins are what make up our cell membrane. I spoke to you about transport at the beginning of the lecture. Trans means across. So we're talking about materials moving across a space. There are two kinds of transport that cells are going to use. The first we're going to talk about is passive transport. And passive transport requires no ATP, no energy from the cell. Think about passive people, they're just mellow, and they don't put a whole lot of energy into their day-to-day. Now, three types of passive transport can be used by cells. The first is diffusion, okay? And diffusion is when we take materials from a high concentration and they pass across this membrane to a lower concentration. Now, over time, you're going to see equilibrium be reached. And equilibrium means we have about the same concentration of molecules on each side. So I'm going to show you diffusion real quick here on my desk. I have water and I have food coloring. So if I drop my food coloring into the water. Then you can see a high concentration in the cloud there. And I'm not putting any energy into this. I'm not stirring it up. I'm just letting it flow. And we'll look at this again here in a few minutes, and you'll be able to see that this, this food coloring is going to diffuse throughout the water. Another type of passive transport we have is called osmosis. And osmosis is the diffusion of water. So water is moving down its concentration gradient from a more crowded condition to a less crowded condition. And that's what we saw with the eggs. The water egg took in water because there was more water outside than inside. And our syrup egg lost water because there was more water inside of the cell than outside, so it flowed out. We also have facilitated diffusion, and facilitated diffusion is like diffusion, but it uses a protein. Okay, so diffusion itself would just be squeezing through these phospholipids. Facilitated diffusion is going to use a channel protein or something called a carrier protein. So I want you to notice that these little green hexagons, there's not a whole lot of them in the intracellular space. This would be in your cytoplasm. But there's a lot of these little green hexagons outside of this cell. So we can see that they are going to flow from their high concentration to their low concentration through this protein, and that is facilitated diffusion. We also have these little light green ovals. There's very few of them in here in the cytosol or in the cytoplasm. There's a lot more of them out here outside of the cell. So we can see this carrier protein is taking in these molecules and releasing them on the other side. So they are still going down their gradient. Make sure that you pay attention with facilitated diffusion to the protein and which way the molecule is going. And we'll talk about why in just a few minutes. Now I want to look back at our water. You can see now that we have pretty much an even distribution of this green food coloring throughout our water. So this would be an example of diffusion. No energy required to make that happen. Now I mentioned that diffusion does not take energy in the form of ATP from the cell. Instead, it's going to depend on the random movement of molecules, and molecules and atoms are always in motion as long as temperatures are above absolute zero, which is negative 273 degrees Celsius. We're never going to reach absolute zero unless maybe you're in deep space somewhere. So these molecules and atoms are constantly going to be moving. Even in solids, they're going to be lightly vibrating all the time. Now these materials are going to constantly collide into each other, and they're going to spread out. And we can see here we have a high concentration and a low concentration. So these molecules are going to bump into one another and send each other across this plasma membrane where they have more space to move around. Now you can see we're building our concentration in time until we reach a state called equilibrium, where the concentration on both sides of these membranes is about the same. So that means our molecules are going to be fairly evenly spread apart. Now, here's the key to equilibrium. Remember that molecules never stop moving. They're going to continue to bump into one another. So molecules are going to be bumped back and forth across this membrane, but it's going to happen at a fairly equal rate. For every molecule that's bumped to the outside of the cell, one is going to be bumped to the inside of the cell. So we maintain this equal concentration. The membrane is said to be selectively permeable. Only certain molecules are going to be allowed to pass through. Permea means to pass by. So permeable means that a substance can pass through it. Impermeable means that a substance cannot pass through it. Think about rain falling on a dirt road. The dirt is permeable to the water. It will allow the water to go through it and get into the ground. But asphalt is impermeable to water. When rain falls on an asphalt road, you just get puddles because the water can't soak through the asphalt. Now, molecules that can move passively across the membrane are water, glucose, carbon dioxide, and oxygen. CO2 and O2 are very small, 3 atoms in this one, 2 atoms in this one. They're also uncharged, so they can squeeze right through the phospholipids in that plasma membrane. Water has a charge. It is going to need a protein to go through the membrane, but it can still move down its gradient, just like we saw with the eggs. Glucose is large, and it's going to need a special protein to take it through the membrane as well. Now, all four of these are going to move down their concentration gradient, which means they're going to go from a high concentration to a low concentration, from where they are more crowded to where they are less crowded. Now, I want you to imagine going into your favorite fast food place, and you're with your buddy. You both get a drink. You like ice in your cup, so you're going to fill your cup full of ice, and then you're going to get your drink. Your friend doesn't really like ice, so they don't get any ice in their cup. They just fill it full of drink. Who has more drink? Hopefully you're thinking, well, your friend, because the ice is taking up space in your cup and there's just not as much room for the drink in there. It's the same thing when it comes to solute in a solution. If we have more solute, it's like the ice. It's taking up space, so there is less space for water. When we look at this picture here, you see an even level of liquid, but there is no salt on this side, and there is salt on this side. Okay, so the salt is taking up space, therefore there's less water over here than there is over here. Therefore, this water is going to move down its concentration gradient and fill up this side of our cup. Okay, you can also think about this like salt and sugar suck. Wherever there is more salt or sugar, there is going to be less water, and the water will move in that direction. Tonicity is the ability of a solution to pull water out of or into a cell. Okay, so there are three types of tonicity, and we examined two of these with your egg lab. So hyper means above, hypo means below, and iso means same. And all of these are referring to the solute concentration, salt or sugar, that is present outside of the cell. So a hypertonic solution has a higher concentration of solute outside of the cell. We saw that with your syrup egg. There was more sugar outside of the cell than inside of the cell. Therefore, the water was pulled toward the sugar where there was less water, and the cell shrivels. You can see that here in this picture with blood cells. Hypotonic solutions would be like the water that you place the egg in. There was a lower concentration of solute outside of the cell compared to inside of the cell. The cytoplasm has salts and sugars in it, but the water did not. So the water was going to move inside of the cell, and we saw the cell swell. Now, this can possibly lead to a cell bursting. You can see that here with these blood cells. Okay, see this one has lysed or burst open. Isotonic solutions are when solute concentrations are the same on both sides of the cell. We did not have an isotonic solution in your egg lab, okay? We used air as a control, but this was not an isotonic solution. We were not able to set up a solution where both concentrations were the same. However, if this happens, you're going to get molecules bouncing back and forth across that plasma membrane at the same rate, so you'll be in equilibrium, and there will be no change to the cell. You can see that here with these blood cells. This is where we want to be when it comes to your blood. Okay, we want to have that blood be isotonic to the blood cells. I've included a video here to kind of summarize what happened with Jennifer Strange. These are actors. This is not her. This is not the actual contest. But if you pull up this PowerPoint and you maximize it on your screen in the presenter view, you can click play and it'll take you right to this video. And it will tell you exactly what was going on inside of her body as she drank all of that water. I'll let you watch this on your own. I'm not going to play it here. We talked about passive transport, how cells move materials across a space, across a membrane, without using cell energy in the form of ATP. The other type of transport cells are going to use is called active transport. And active transport does require the input of ATP energy. Think about active people. They're always on the go. They're always using their energy. Now, active transport works backwards from passive transport. In passive transport, we were moving materials down their concentration gradient from where they were crowded to where they were not crowded. Now, active transport is going to do the opposite. It's going to push things back to that crowded side of the membrane. Now, you can think about water in the lake and water in a little segment of river right below the lake. You're going to have more water in the lake than you do in that little segment of river, so the water is going to freely move downstream into the river from the lake. If I want to push that water back into the lake where it was more crowded, I'm going to have to use a pump. And cells use pumps as well in the form of carrier proteins. So here's a couple of examples of this. First, I want to look at these little purple molecules. These are salts. Okay, you see a lot of salt outside of the cell, and yes, I know this is not in English, so just listen to me. So we have lots of salt outside of the cell. We have very little salt inside of the cell, and we want to keep it that way. So we have a pump pushing materials, this salt in this case, outside of this cell, and it's using ATP or energy in order to do this. Now, we also have this little green hexagon here. This is glucose. You can see there's not much glucose outside the cell, but there is glucose inside of the cell. But we want to get that sugar into the cell so that we can use it for energy. So in this case, we're using this salt that has built up outside to pull this glucose in with it as it passes through this protein. And we'll look at this a little bit more later. I'm not concerned that you know these two types of transport. This is primary, this is secondary, but I do want you to understand that active transport pushes materials against their concentration gradient. Carrier proteins, like you saw on the last slide, move materials against their concentration gradient. And in order to do that, the substance or the material needs to bind to the carrier protein. And when it does, it'll change the shape of that carrier protein and allow the substance to pass through it. Okay? Now, there was one protein pump that we briefly saw on the last slide that I do want you to know. It's a very important one. It's called the sodium-potassium pump. Okay? And we can see it here. So let's just walk through what the sodium-potassium pump is doing. It's very important that we keep that sodium outside of the cell and keep the potassium inside the cell. This is going to allow nerves to function properly. So we see here our sodium that is inside the cell. It's building up, and we're going to bind 3 of those to our carrier protein. ATP is going to come in. This is your cell energy, and the combination of these 2 things will change the shape of this sodium-potassium pump and open it up to the outside. So this sodium is now free to leave, okay? And this potassium is free to enter, and we can take 2 potassiums into this carrier protein. Once this happens, your phosphate is going to come off of the ATP, and this is going to allow this shape to change back to the way it was before, and this potassium can then be released to the inside of the cell, maintaining these 2 gradients where they need to be for this cell to function properly. If we think about the sodium-potassium pump, I want you to hold your hands like this and think about this side as being the inside of the cell and this side above your arms as being the outside of the cell. So here we're going to take in the sodium and take in potassium. Take in the sodium, release the sodium, take the potassium, release the potassium. Take the sodium, release the sodium, take the potassium, release the potassium. And it's just going to go back and forth like this in order to maintain these gradients, which are super, super important. Not only does this have to work right for your nerve cells to function, but nerves are going to control your muscles. And also there's a lot of other molecules, like we saw that glucose on the last slide, that will hitch a ride with that sodium as it diffuses back into the cell. Homeostasis or balance is very important to maintain, and temperature is one of the things in an organism that must be maintained at a certain level. So is pH, because both of these factors control the rate of biochemical reactions through enzyme activity. So we have a graph here showing you enzyme activity at different pHs. The blue enzyme has an optimal activity around a pH of 3. The orange enzyme has an optimal pH of around 7, where it's going to get the best activity. And the green enzyme has an optimal pH of around 11 or so. We could make a similar graph like this with enzymes and temperature, especially with different organisms that have different body temperatures. Their enzymes are going to function best at whatever their body temperature might be. Now, all single-celled organisms and many multicellular organisms like snakes, fish, and frogs depend on the external environment in order to maintain their temperature in their bodies. Now, these organisms have adapted to survive temperature swings because, as we know, the temperature is not always the same outside. So mammals can go through hibernation, where they lower their metabolic rates, they lower their energy use throughout the winter. Estivation is when mammals hide away from heat in order to conserve energy and water loss. Mammals and birds are warm-blooded. They can control their temperature internally through different mechanisms. And reptiles, like snakes, for example, might lay in a sunny location in order to warm themselves up and then move to a shaded location in order to cool down, or may even put parts of their body in the water in order to cool down as well. Now, if you've ever seen a snake in the summertime, they can move pretty fast. Their enzymes are working at the proper rate because they're at the right temperature. But if you've ever found a snake in the winter, then you notice that it can hardly move, if it can move at all, because its enzymes are not at the right temperature and it's not able to react fast enough to do its daily functions. All right, that's it. These are my resources. Hopefully that made your egg lab and homeostasis and transport in cells make a little more sense to you. I will see you next time.